This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 512, recorded on September 21st, 2018. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
Pretty cloudy out there, isn't it?
Yes, it is. All the way out to Penn State University, which is where I just returned from.
Back from there?
Drove back this morning, and lovely drive. It's truly a named state that keeps its name. It's called Pennsylvania. It's Pennsylvania. It's a gorgeous state. It really is beautiful.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here, but it's overcast here, 69 degrees, 21 C. Kind of cool, maybe thinking about becoming autumn one of these days sort of weather.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Hey, Rich.
Rich.
Got 85 degrees, 29 C, pretty much overcast, probably a little more rain on the way, which is good. We've had rain on and off the past few days. It's all good.
Okay.
It's all good. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. I was excited when I heard Vincent and Dickson and Alan talk about their temperature. I thought I might be the hottest, but I forgot about Texas.
No.
So we're up at 71.
Oh, wow. You were
71. What do we have here? We have 21 Celsius.
So I'm 22 Celsius here, but very cloudy, as Dickson would have seen on his drive by.
Did you drive by Madison?
No, I drove up Route 80. So Route 80 doesn't go by Madison.
And joining us today from College Park, Maryland, Ann Simon.
Hi, everyone. I'm so excited to be here.
Welcome back. You were here. You weren't here. You were on TWiV back in July. Wow, it seems like forever ago, July. Yeah, in terms of weather, it was forever ago.
Exactly.
Yeah.
So we promised we'd have Ann on about once a month to talk about plant viruses.
Wonderful.
Yeah.
So welcome to TWiV. And so we have a big crew today.
I'm excited to be here. Do I get the weather forecast from College Park?
You bet.
Sure.
Actually, yesterday it was a balmy 89.
Wow.
Ooh.
And today it is a very beautiful 79. And I just want to let everyone know that I am sitting here in my office. I do have a puppy on my lap. So if you hear barking or whining, it is not a commentary on what anyone is saying.
I thought you were going to say, if you hear barking or whining, it's not me.
I thought you were going to say I have a puppy in my lap, so I win.
Okay. We like those kinds of sounds. That's great. But 89, see, that's the difference between having a sun out and not, because if we had no clouds today, it would be in the 80s, right?
Maybe.
And I'm not ready for this 21, 22 weather yet, but who cares about what I want, right?
It's not about you, Vincent.
It's not about me.
Or me either.
We have some bingo contest results. Remember, a couple of weeks ago, I said, if you get a TWiV bingo, you'll get a copy. The first 5 people will get a copy of Principles of Virology. So we have 1, 2, 3, 4 entries. And each of them actually, so Alex wrote, I think I have a bingo. I originally thought when you talked about it, it was only for episodes after you released the card. I later found out we could use older episodes and went to download a long list for a long trip that happened to land right after I finished. And so Alex put down some of the things he got and the timestamps as well as the episode. For example, we have a show title, episode 495 at the end. That was the first bingo I found, but while listening to episodes, Vincent was mentioning how he got an email from someone talking about chainsaws. I heard you talking about the story arcs. Okay. I like listening to the backlog, but doing multiple episodes in a day has given me a real appreciation of the time that goes between episodes. And then Eric wrote, I think I've got bingo. I hope other listeners play too. It didn't take all that much time. Plus, it gave me an excuse to go back and listen to some classic old episodes.
Wow.
So, for example, one of them, Plaques Are My Life. I Love Plaques. Episode 68 at 7 minutes and 18 seconds.
Sound like a dentist.
And the title of that episode was Ode to a Plaque, so that was not a hard one to find.
Here's one for Anne. Cucumber mosaic virus hacks aphid behavior to maximize its spread. Dickson laughs.
True.
Wow.
Episode 70, hacking aphid behavior.
So you have had plant viruses on TWiV.
Yes,
We've talked about them there. Yes,
for sure. And in fact, after our episode at ASV, someone wrote in and said, I think it was Steen Hoyer, who you may know. He's a postdoc with Siobhan Duffy. He said, he gave us a list of episodes on which we talked about plants. And he said, I know that Anne was just kidding. The first plant episode after 500. So Jolene wrote, Dear Twivites, I'm writing to show you I got a bingo. It was harder to complete than I expected. It wouldn't surprise me if others need time to listen to old episodes. I'm wondering if something like this could be used when implementing podcast episodes in a class as a way to guide students in their listening.
You mean to check and see that they listened?
Of course.
Yeah.
So Jolene sent in a picture where she put the TWiV episodes on the bingo card.
Good.
And she got a vertical bingo. She put a lot of other ones in, but you know how it is. You put lots of chips on your card, and then all of a sudden you get a bingo. So that's good. And she has a listener pick, which we'll do later.
I don't remember anything about I hate palm trees. Did anybody get
Yeah, me.
That's
me. That's me.
Oh, okay.
I don't really hate them, but
we've had some interesting discussions about angiosperms also.
It's funny that no one got I hate palm trees, but when I was talking about it's a long story we won't go into. Nicholas writes, hello, viral communicators. I'm a grad student at UW-Madison, long-term listener. When I heard that Vincent's contest was for principals, I thought I had this in the bag. Boy, was I wrong. I spent over 40 hours going through old shows.
Wow.
Even worse, I found that while I had a memory of the phrase, I couldn't figure out what episode it was in. Fortunately, I can listen during lab work. So he sent a bunch of bingos. Did you get your flu shot? Episode 470. Right. I hate cherry trees, 488, etc.
Who said I hate
Vincent
cherry
hates
trees?
cherry trees.
I don't like trees.
So, Anne, if you're wondering why we don't cover a lot of plant viruses, it's because Vincent hates so many different plants.
Yes.
Palm trees and cherry trees. Here's a fun story. One day at lab, my PI...
Wait.
Yes.
Here's a fun story that will make the plaque pack happy. I like that. Plaque pack.
Go ahead, Rich. Read it.
No.
Yeah,
okay.
Go ahead.
First, a fun story that will make the plaque pack happy. One day at lab, my PI asked me how a plaque assay works. I should note that I work in an Epstein-Barr virus lab where the virus is quite content not killing itself, thus making a plaque assay impossible. So this could have been a much more difficult question than it first appears to other virologists. It had been quite a while since I had taken my first basic virology course, but fortunately I had recently listened to Ode to a Plaque, TWiV 68, which, by the way, is my all-time favorite episode. And I was
could have guessed.
And I was able to answer this question in exquisite detail. I will now always know how to conduct a viral plaque assay, and I will never consider qPCR for viral genomes a substitute. I cannot thank you folks enough for doing this podcast and sharing the collective knowledge each of you have accrued over the years. As long as this wonderful group keeps podcasting, I'll keep listening. And he's got a listener pick, and he's got a bit about chronic wasting disease here. Did you move his listener pick somewhere?
No, it's there, but we'll read it later.
Okay.
We can read it. We'll read the chronic later also.
Okay,
and by the way, Dickson, he wants you and I to sign the copy.
What, of the virology?
Yeah.
Really? I would be an imposter if I did that. I don't.
All right.
I'm not.
Yeah, well, it's okay. You've always been an imposter.
I beg your pardon.
Come on, Vincent. I mean, Dickson, you're a celebrity, okay?
So
you're a virology celebrity.
Nicholas, Jolene, Eric, and Alex, send me your addresses TWiV@microbe.tv. And tell me if you want a PDF or a hard copy. I can have both. They're both the same price. They're both $190.
How about that?
So you're getting this because you played and everyone else didn't, so they don't get it. And PDF or hard
play
copy.
To win.
Can have either one. And finally, we have an email from Tom who writes a rant. I urge everyone listening to this podcast who hasn't yet done so, immediately set up a monthly donation to TWiV. You probably don't think twice about adding a $1 candy bar or $1.50 soft drink to your weekly or bi-weekly grocery shopping cart. Do the world and your health a favor, set up an automatic Patreon contribution of that money. And when you're standing in the checkout line, remind yourself that you have already used that money to do something good for many and not do something bad for yourself. Vincent, feel free to read this rant on all the other podcasts. So I have been looking forward to future TWiVs. Tom in Austin, who we met during 500.
We did.
Thank you, Tom. Appreciate it. And I put it up front so people will hear it, because by the time we get to my rant at the end of every episode, no one's listening anymore.
Arg.
Okay, let's talk about plant viruses. This is all plant viruses. You know, Ann, on every show we have, unless it's a road show like we did with you, we have a snippet and a main paper. And today both are going to be about plant viruses.
I'm floored.
And you picked
so
both
excited.
of them.
I know.
So the first one is plumpox virus.
Yeah,
tell us about plumpox virus.
So plumpox virus is this absolutely horrible virus. It's a member of the podiviruses, and it is busy decimating all of the nut trees in Europe. So it is a huge agricultural problem in Europe. Just cannot be any worse. And of course, we have a major industry here. People like their apricots and their peaches and their almonds. And so the U.S., ever since it's been, running rampant through Europe, has been very, very worried about it coming to the USA. And it's in Canada right now. It's across the Niagara River. And it's so widespread in Canada right now that they've stopped even trying to control it. So pretty much their industry is going to be gone very soon because this virus kills the trees. And then there's us here. Big country, lots of nut trees. And it's so interesting. Lots of nuts, yeah. and nut trees.
Does this kill the hazelnut trees also?
You keep referring to nut.
I think it pretty much kills just about all of the nut trees, but I'm not an expert in this particular virus, but I just know that
you're referring to nut trees, but it also kills peaches, plums, apricots, stone fruit, right?
Yeah, stone fruit as well.
And
so
then,
nuts and
wow,
that's huge.
So it's really big. And the podcast where we talked about citrus greening, which was the 503. I mean, that's a huge problem in citrus. It's killing all the citrus trees worldwide. Not a virus, it's bacteria. But this is a virus that's just devastating Europe. And what was interesting is that in 1999 in Pennsylvania, a very observant grower noticed a very odd, I think it was an apricot tree. And he thought there was something wrong with these trees. And fortunately for everyone, he asked a plant virologist, a plant pathologist, to come out from Rutgers University to check on the trees. And there's extension people at all of the universities and all of the land grant universities in every single state with people who will come out and look at problems like this. And this one guy, Jerry Freecon, went out there and he looked at it and his heart must have just dropped because he said, I think this is plumpox. And it was the first case of plumpox in the country. And $30 million later, they managed to contain it to a few trees in the county in Pennsylvania. But literally, what do you do? How do you quarantine? They destroyed every single nut and stone fruit tree within about, I guess it was about maybe as much as 500 meters of that tree. They all had to go because there's no cure.
How does it spread?
So plumpox is spread by aphids.
So it's a vector-borne disease.
So it's vector-borne, like virtually all plant viruses are vector-borne.
Yeah, exactly.
And so you don't know how far the vector's gotten, and it takes years and years and years before a tree shows symptoms. So then they tried to figure out how these trees got infected, and the farmer had done everything correctly. He'd bought it from a nursery stock, and the nursery had certified it was free of viruses. Yeah, right. And and it ends up with plum pox. And so, ever since then, they've been, worried about plum pox virus. And sporadically, trees come down with plum pox virus. So the most recent one was in 2015 in New York. So they had a tree in Michigan come down, and they've had a couple of trees in New York. And so the latest one, it's in the Hudson Valley, 2015, and they've been, surveying all the trees. So this is, I actually talked to the woman at APHIS. I should have written down her name, but she is in charge of keeping plum pox out of this country. And what APHIS does, this arm of they've got an investigative arm, and then they have an arm of these. APHIS is they control pathogen entry.
Right. So this is the Animal Plant Health Inspection Service. And actually, if you've traveled to certain places like Puerto Rico and I think Hawaii as well, you probably had to go through an additional screening, and you got a sticker from APHIS on your luggage.
Ah, yes, that's
yeah. So they're the people who are responsible for keeping stuff like this from coming into the country.
What they do is they send out teams, and they knock on every single door within mile radiuses of an infected tree, and they ask to look at the backyard trees and see whether or not they can find any others. And some homeowners are like, oh yeah, sure, come look at our trees. And others are, no, go away.
What about walnut trees?
Same thing, walnut, yeah.
Because they're all over the place.
Yeah, that's
But maybe
true.
they're not susceptible to aphids. Who knows?
Well, that's the thing, is that if the aphid doesn't like the tree, it's not going to transmit the virus to it.
Right.
I think about the almond industry in California.
Well,
they're very worried.
They're in trouble already. So,
Anne, you said there was this Pennsylvania, then Michigan. Are there other known U.S.?
Just New York. So New York's had a couple of cases over the years, and the last one was 2015, and it's still quarantined. So the area you can send fruit out of the area because the virus doesn't get into fruit. And I should mention for the listeners that it's perfectly harmless to eat fruit with plum pox virus or any plant virus in it. In fact, peppers, we eat, pepper viruses all the time, and every potato you eat is filled with potato viruses. So they're not harmful at all to people, but it's very harmful to the tree. And trees, they live such a long time, and they're so valuable that it isn't like it gets into a crop and, okay, you lose that crop or you lose just that field one year. But the trees, you just have to cut down these trees. And so USDA, they wanted me to mention to please let the APHIS people inspect your backyard
Yes,
trees.
sure.
They've got uniforms on. They don't want to do anything. They're not there to say, oh, you've got an infected tree. What did you do? No, they just want to make sure that they can get rid of any infected trees. And so since 2015, they've been clear for 3 years. They still inspect every single tree in this huge radius of where they found it. Every couple months, they take samples of every single tree, and they have 3 more years of this. And then they'll say, okay, it's not back here.
So
what's the cause of death, by the way?
Yeah. Right. How does it kill a tree?
Oh, how does it kill the tree?
Mm-hmm.
What usually happens is the tree mounts a defense. And trees are not really bright about how they do this.
Stop! I'm so disillusioned now.
I know. I'm sorry. But these trees, they think that there's some horrible disease, infecting them, and so they mount this massive defense to kill off the disease, which they manage to do by killing themselves off. So, the tree dies.
What do they do? Secrete more sap or make a gall?
Yeah, they secrete callus into their veins and
xylem and phloem.
Right.
Yeah, because viruses travel through the veins.
What's callus? I don't know what that is.
Callus.
Callose.
So callus, it's this gummy stuff.
Gummy stuff.
Yeah, gummy stuff. So it kind of gums up the phloem, which keeps the virus from being able to travel, but it also keeps all the sugars and nutrients and everything else from being able to travel, and the tree dies. But a lot of the reason why why plants become symptomatic is because it's the defense that they're mounting against the pathogen.
The aphid presumably bites a stem way up in the leafy part of the tree, right?
And they bite the leaves.
The leaves. And then does the virus spread throughout the entire tree, up and down, from root to top?
Yes. So podoviruses they're viruses that are limited to just the veins. This isn't one of them. And so this virus just spreads everywhere. So it gets into leaves. It just doesn't get into the fruit, apparently. Mm-hmm. But it gets into leaf. It gets down to the roots. And so it kind of gets everywhere. And the tree's desperately trying to kill off the places where the virus is. And it ends up killing itself. And
so are there any
but
strains?
It's a slow death.
Are there strains of these fruits that have developed resistance?
So not really against plum pox. But this is a big deal right now. There are other viruses. We're going to talk about geminiviruses next, and they are definitely developing strains that are resistant to these geminiviruses. And what's interesting is that they're actually sending these plant strains to other countries as well, because if they can control the virus in other countries, then they can hopefully control it here. But what aphids just can't stop is people saying, in the old country, I had this wonderful plum tree and I want a little piece of it in my new home in America. Send me a branch, send me a little cutting of this tree that I can now grow it in my own backyard. And they send it through the mail, and that's enough. That'll do it.
Do they know how the virus was introduced in any of the cases in the United States?
Yeah, they can actually trace it back to people getting it in the mail, and they can trace it back to nurseries. So APHIS has their own investigative branch, Mueller has nothing on these people, boy.
This is a secret branch of the government, so to speak.
It's a secret branch. They try to trace back years and years and years of where things actually came from. And in a lot of cases, they can. So as I say, we're going to talk about geminiviruses that are just these unbelievably awful viruses as well. And they're rampant in China. And the way China ended up getting the Gemini viruses was they imported all this hibiscus for the Olympic Games from India and Pakistan.
You're kidding.
And it was chock full of this virus, of a virus. And that virus is now swept through their cotton and their all sorts of their okra and it's just this terrible virus that's out there. But yeah, it was in imported hibiscus. So for California, getting trying to import a citrus variety from another country can take 2 years of testing to make sure that this planting that they've gotten, this young tree, is actually free of everything. And they confine it. So it's kind of like, but more severe than England. You want to get your dog in England, you have to put him in quarantine for a long time. And that's what they do with these trees.
So if the fruit of the plant doesn't get infected, why don't they just start from seeds?
Well, they do. I mean, but it takes
They should be free of the virus though, right?
Well, yeah. So in my lab, I'm growing some orange trees and lemon trees from seeds. Right. So we planted them about 3 months ago. They're about 1.5 inches tall.
I got it.
Well, the other thing is
they have about 6 leaves, and they are getting TLC.
I'll bet.
I mean, great lights and lots of water. And
how long does it take to grow an orange tree, for instance? What's the
many years?
Many years.
Many.
Really?
And to grow a tree. And
the other issue with this is a lot of fruit trees don't breed true from seed, right?
Right. So you have to graft. So you have rootstock, and then you have the top part of the roots, and so you have to kind of splice them together. And so yeah, I mean, it's a big deal. And then you graft other things onto them, and it's a big deal. The trees take a long time to grow, which is why if you find a resistant tree, I mean, I don't think there are any like wild orange trees, but if you find some resistant tree, but it doesn't have the characteristics of the fruit that you want, you have to then cross it to ones that do. And that's a big problem because then it's years and years and years, and then you have to cross it again. So it's not like, Drosophila that, what is it, 9 days or something? Or
That's quick.
Oh, it's quick, yeah. Or, Cinerabdupes, or very, very quick.
Yep.
You're talking decades. And so this is why, no one's working, or maybe they are working, but they're going to work a long time on trying to make genetically engineered, because we can genetically engineer trees to be resistant to this virus. There's no question about that. That's simple. That's very simple to do. But actually, getting the trees grown then and with the right properties, and then making people understand that it's perfectly fine to eat it is another thing.
What's the basis for the resistance in those plants?
So it's all RNA silencing.
Well, what about grafting one of those onto a diseased tree? Would that spread then?
Yeah, so you could but you still got the same problem, is you still have to back cross. You still need the fruit to be right.
No, I mean, could you cure a diseased tree with a small branch of one of the genetically engineered plant?
No. So
it stays put. The RNAi stays in the cells. It doesn't get distributed.
It not as much as it needed to. I doubt that could actually happen.
Just an innocent question from an innocent bystander.
No, that's fine.
That's a good question.
I did start out as what we call a plant person, and so I work on turnip and tobacco, and so we don't have any problems. It grows, it dies. Arabidopsis, it's fast. Yeah. So this is all kind of new to me, the whole citrus thing. And
so, Anne, you sent us an article about a papaya virus, papaya ring spot, and that was dealt with by making resistant plants, right?
Yes,
in Hawaii, but it must grow faster.
Gonzalez, right? He is the savior of papaya in Hawaii. And they made resistant plants and resistant trees, and now they can grow papaya because the virus was such a scourge. It killed all the papaya trees.
But they grow a lot faster, I guess, right?
They grow faster.
Yeah.
My guess is they grow faster, but still it took them a long time to develop it. And I don't think that they have quite the same characteristics that people like about oranges.
Mm-hmm.
I mean, I'm not a papaya person. Maybe you would know, but it doesn't really matter what the color is, or the thickness of the skin, or the number of seeds, or the sweetness, or the pulpiness, or I don't know.
Or the amount of papain.
And could you really briefly elaborate on exactly how they're engineered to do the silencing?
Yeah, so what happens is they actually just take a piece of the virus. So they used to take a piece of the region that coded for the coat protein of the virus, and the coat protein is what makes the that encapsulates it. So it's the particle of the virus itself. But then they realized that they could actually take any piece of the virus. It didn't matter if it was the coat protein piece, and they just take that piece and they make a plant that is expressing that piece, and that piece gets chopped up into little RNAs, and those little RNAs are like a surveillance. They hang around, and if the virus gets in, the little piece pairs with it. It sticks to it.
They're like aphids.
Hybridizes to it, and it and then the RNA silencing system. There's an enzyme scissors that comes along and cuts it, and it just cuts the virus into pieces.
It almost sounds like
sounds like CRISPR, doesn't it? Because it's very similar to the CRISPR system. So
what's the potential for a virus overcoming that resistance? Does that happen?
No, because you put a big piece of the virus in.
Okay. So this is a pretty solid cure.
It's very solid, and it works great. And so there are a number of plants that are now completely resistant to the viruses. And this is especially important for like the geminiviruses that I'm going to talk about, because these are very, very severe pathogens, and they cause billions of dollars in losses. And it's such it's an easy fix. I mean, anyone can. I mean, not anybody, but it's pretty simple to do now. We've had this technology a long time.
A caveman can do it.
Caveman.
Right. So it's easy. It's simple, but not easy.
So
got it. Straightforward.
I can do it. So let's put it that way. But yeah, I mean, plants don't. Again, plants. They're not that smart. And so, they don't have a circulatory system. They don't have antibodies, darn.
You're going to get mail from the plants. I mean,
What are you saying? We're not very smart.
But so all they have is this RNA silencing system. I mean, that's their protection against viruses. But viruses have figured it out. And so they make a protein that protects them against the RNA silencing. So if a plant is already producing the RNAs, there's really nothing that the virus can do. But if the plant is not producing these little RNAs, then the plant virus comes in, it makes a protein which disables the RNA silencing system. And now the plant begins to look kind of weird because it needs its silencing system just to develop normally. And then the virus can just take off and do whatever it wants. So it's kind of it's like a race between the virus and the plant, and the virus usually wins. So,
but the engineering gives the plant a head start, and then you're
absolutely
golden.
So it's very simple.
Got it.
It's completely safe, and so that's the genetic engineering alternative, the GMO. So very safe, And what do you do if you're not doing the GMO? You have to control the vector.
Right.
So what you have to do is spray and spray and spray all these poisons all over the plant that kill every insect,
Yummy.
that kill all the birds that eat the insects, that kill all the things that eat the birds that eat the insects. And so it's really an environmental disaster.
Yeah.
But if you have the GMOs, you don't have to spray at all. So farmers love it because they don't have to spray. It's much safer for them. And the problem is that there's, a lot of people out there with loud voices who are lying about the safety of this. I mean, it's got a little piece of the virus. It doesn't even have to code for anything.
It doesn't even get into the fruit. It doesn't get into the fruit.
It doesn't get into the fruit. I mean, it's so innocuous to make this extra little piece.
And they don't trust you. That's all that is.
I know. And I'm
well, but the other issue is, in order to get it to that point, it's, as you said, it takes a very long time when you're dealing with a fruit tree, because you put the gene in, and then you have to grow up the tree and get the fruit. And
it's just not a solution for trees.
Yeah.
So
it's a solution for crop plants, annual.
Right.
It's not a solution for trees.
You said this virus takes a long time to kill the plant, the trees. So what is the age of a plum tree that lives a disease-free life versus one that got infected? What's the big difference? I mean, is it 10 years, 100 years, 5 years, 3 years?
I know that the orange trees can live, 75, 80 years. I mean, olive trees have been around.
Forever.
They must live hundreds of years.
Exactly.
Right? Exactly. So I don't know about the nut trees, but certainly, you're talking decades and decades and decades of life. So these trees are very valuable, and they produce a crop every year that's worth a lot of money.
This is true.
And then it gets infected by this virus, and there's no cure. There's nothing they can do. The only thing you can do is cut the tree down and desperately try to not have this thing spread, and try to, they surveil aphids, they surveil all these other trees. And
there's a viral infection in pigs called hog cholera. And when an outbreak is found, within a 100-mile radius, every pig dies.
Yeah, it's the same thing with the trees, and that's a big.
It's not a very good solution.
I'm sad for the animals.
It's caused by a pox virus, right, Rich? Hog cholera.
Which hog cholera?
Another pox
classical
virus.
Swine fever.
I'll be darned. That's a big problem.
I'm not so sure that there's African swine fever. That's a pox virus, but I'm not sure about hog cholera.
Okay, African swine fever is the pox virus. That's what I'm thinking about.
I think I don't think that's a pox virus.
Classical swine fever or hog cholera or Schweinepest.
Right.
I love that.
It's only because of that virus, though, that the farmers are obliged to boil the garbage before they feed it to the pigs. It's not for trichinosis
it's a pesty
control.
virus.
It's a pesty virus.
Is it?
Which is in the family Flaviviridae.
Is it a vector-borne?
Aren't they all?
I thought
coming from the plant virologist here.
It's okay.
I bet they're not vector-borne. It's probably pig to pig. They do diarrhea, and it's a mess in a pig pen, right?
It's a mess.
So I would think it's not. Now, Anne, why do people, do you have, we talk about anti-vaxxers all the time, and there's anti-GMOers, And we don't understand why anti-vaxxers are anti-vaccine. Do you understand why anti-GMOers are such?
I think, well, the thing is that there's a lot of voices who are very anti-large, companies,
Right.
the large pharmaceutical companies that think that they're just doing things to get a buck.
A conspiracy theory.
And but there's also, these businesses have to make money. It's also true. And so if you're against these large, these large companies and you're for the small farmer, one of the things is that some of these plants that were being produced, these GMO plants, they grow better. They don't need, it's cheaper to grow them. But the seeds, you have to buy the seeds every year.
Sterile. That's right. That's right.
But they're only sterile because that was the huge thing is that, they're propagating across fields and stuff. So then they made them sterile. And now the outcry is, they're sterile. And so then you can't.
Exactly. You don't win on this one.
you can't win. No matter what you do, there's always, that goalpost keeps getting moved.
No question.
But I think it started with this. I think it started because also people didn't understand, and I think the industrial people weren't taking the time to really just explain to people exactly what was going on. They just said, no, it's safe, it's safe.
I think there's another aspect on the industrial side, though, which is that the dominant company in the field, Monsanto, has behaved kind of the way you would expect a vast monopoly with a stranglehold on an industry to behave. They haven't always been, they haven't always been good PR for themselves. Let me just leave it at that.
I didn't think they had to. That's right.
Yeah. I mean, they, and Monsanto became such a giant in the field because they had the best herbicide.
Right.
They had an herbicide that was so much safer than all of the other herbicides that are out there, which is glyphosate.
Yeah.
And so then they made plants that were, the only problem with glyphosate, because they've been using it since, forever, since way back in the last century, because it's so safe. And, but the problem is, you couldn't use it during the growing season.
It's non-selective.
Because it's non-selective. It just kills anything green. And then during the growing season, you had to use these other herbicides. So they made their crops resistant to glyphosate. And then all of a sudden you could use it all the time. So farmers liked it because it's a very safe herbicide and it works very well and it doesn't build up in the soil or anything. But that gave Monsanto this edge. And,
this is Roundup, by the way, right?
This is Roundup. Which is what I use at home because I don't want to, poison my puppy here. So we got my puppy on my lap who's sitting here sleeping. But I also think another thing that happened was it was the Europeans that really, had a huge outcry against GMO. And I believe, my personal feeling is that's all because of mad cow disease.
Yeah.
There are two things. I watched this happen right as I was becoming a science journalist, and there are two things going on. One was the mad cow disease. The other, which is not widely discussed, is Europe has had a tremendous desire to protect their farmers from cheap American imports. And American farmers adopted GMO technologies almost as soon as they were available, and so American corn and so forth were largely going over to GMOs. And it became very apparent to activists in Europe that French farmers, for example, were not going to be able to compete with cheap imports of American corn. And because of trade agreements that went into place in the 90s, they were not going to be able to apply tariffs to them because that was being phased out. So how do you keep the cheap American corn from coming in? Well, you suddenly make up a story about how it's toxic.
Yeah, and they did.
This became a huge thing, and they whipped up a bunch of activism around that as a barrier to trade. And so this was a large motivator, I think, for the initial European resistance. It wasn't about the perceived danger of GMOs. It was about protectionism disguised as a perceived danger of GMOs. And then that, because the Europeans were suddenly saying, oh, they're not safe, then people in America said, oh, wait a second, the Europeans are saying these aren't safe. Maybe they're not. So I think that had a lot to do with where we got to be today.
So that's fake news is what you're saying.
Yes. Yes, there was a massive propaganda campaign in Europe that then was imported and infected the United States.
But I think we're slowly turning the tide on that, because some really strong anti-GMO people, now that they understand what it is, have become the opposite.
That's exactly right.
And extremely pro-GMO, because it's so much better for the frankly, to not be spraying these very, very dangerous sprays. And then the organic industry got into it, and they're trying to protect their market by trying to scare people, about their food and saying, no, you have to spend more for our stuff that you treat with poisons like copper sulfate, which I don't want to eat. So I actually never buy organic in supermarkets ever. I won't.
This article that you sent has a nice list of the American grown products that are GMO. It's 94% of soybeans, 92% of corn, 94% of cotton, 95% of sugar beets, 90% of canola oil, and 77% of Hawaiian papayas, among other things. This is just, the industry is just going to wind up pushing us forward, because there's the regulatory agencies are not pushing back against GMO, right? They understand it,
right?
Absolutely.
So it's just going to happen.
Every science society in every country, including the National Academy of Sciences here, are 100% behind it, because it's safe and it is more. It's environmentally friendly. We have potato farmers out here that when McDonald's said they wouldn't buy any more GMO potatoes, they were devastated, the growers, because it was so wonderful. They didn't have to spray. They didn't have to spray for fungus. They didn't have to spray for insects. The potatoes, were beautiful quality, and it was so much more environmentally friendly, and they were able to get much more profit from their crop. And then McDonald's says that, and now they're back to spraying and polluting the Chesapeake again.
You might say they were fit to be fried.
Fit to be fried.
You might say that.
They were.
Alan could have said that. I should.
And this is crazy. It's all this marketing stuff drives me nuts.
It is marketing. Yeah, that's right.
And before we move on, just tell us a little bit about the virus. What does it look like, and what's it made of?
Well, plumpox?
Yeah.
So plumpox is a plus sense RNA virus. So plus sense simply means that as soon as it gets into the cell, it's able to be translated. And it makes one big protein. So it's kind of like poliovirus.
Mm-hmm.
And in fact,
those that's
viruses
the reason. I see, Vincent.
Well,
they're related. They're actually related. They're related.
But the
potiviruses. That's how people who study potiviruses were able to first get NIH money, because they could claim it was kind of a plant, virus like polio. So. You probably don't go along with that, Vincent, but it was kind of like
okay.
it.
But the virus particle does not look at all like polio, right? It's
not at all.
It's flexuous. I love that word. Flexuous.
But apparently, you make a couple of mutations, you can turn an icosahedral, one into a flexuous one, or
really.
What does that mean for us common folk?
Flexuous?
Yeah. Mm-hmm.
It's a big long rod that's looks flexible.
Okay, so this is how I say
you mean like tobacco mosaic virus?
But it's flexible.
My intro bio students is that it's like cooked spaghetti. So tobacco
rubber
mosaic
hose.
virus is like uncooked spaghetti.
I got it.
And this is like cooked spaghetti.
And Ebola is a cooked spaghetti tied in a knot.
That is definitely cooked spaghetti. Yeah.
if you go to
Ebola, this virus is not Ebola.
There's a lovely photo over at Viral Zone of this flexuous filamentous particle, and some of them have two pieces of RNA, right? And
potiviruses?
Yeah, it says here that sometimes they're one or two.
One or two components. That's correct. That's correct. But it isn't like HIV where you have two complete viruses.
No. So the components are separately encapsulated, right?
Right. So it's just been divided up into two different ones.
And they both have to infect a cell to get infection going, right?
They both have to infect.
All right.
That's right.
Are they your favorite poties?
Not at all.
What are your favorite viruses?
I hate them like you hate palm trees. So,
No, that's cherry
I mean,
blossoms.
because
Yes.
because lots of people have worked on them, and it's just one big protein. It's kind of like how interesting. No,
Okay.
I shouldn't say that. People, I don't mean it. I don't mean it to all the plant virologists out there who work on potiviruses.
All right.
It's wonderful, wonderful virus, very interesting, exciting virus. It has a VPG. It's kind of poliovirus-like.
Yeah,
but
it's not a PPG. Yeah, polyprotein.
Yeah, but of course, the most interesting viruses are the Tombus viruses, and
With
that
the
are,
ones you work on, right?
of course, yes, that is true.
All right. Well, you didn't pick one for this episode, but
I did not pick one for this episode because I'm trying to be more universal.
Yeah. All right. So then our next our paper is from Journal of Virology, geminivirus.
I wonder who the editor was on that.
You were the editor, I noticed. And I said, well, I guess she's killing two birds with one stone, right?
Well, I actually picked it to be a spotlight because I just thought this was just an exciting paper. And it's from a Spanish group. And I actually have to, I thought I had it up here, the Spanish group. Eduardo is his first name, but I should get his last name. It's Bejarano?
Bejarano, I think.
Bejarano? Okay. Sorry, Eduardo. I told him to listen in.
Do you know Eduardo? Do you know him?
No, I don't think we've ever met. But this is an interesting paper about a group of viruses called geminiviruses. And geminiviruses are some of the world's worst pathogens. And so just to give you a little background on Gemini. So first, why are they called Geminis? The twins. And that's because they have twinned capsids.
These are so weird.
And it's a little... It's interesting. So there are two types of Gemini viruses. There are Gemini viruses that are two components, two genomes, and Gemini viruses with one genome. But the capsids are always twinned, okay? Whether they have one or two, they have these twin capsids. And these are single-stranded DNA circular viruses. And they're all over the world. There's 360 of these. And they are certainly the most pathogenic viruses. The worst viruses in China, India, Pakistan, Africa, terrible viruses in Africa. They destroy most of the cotton crop every year, most of the cassava crop every year, and most of the corn every year are destroyed by Gemini viruses. And so, again, we have Gemini viruses that are resistant to these viruses, GMO, plants. And they're trying to get, the African countries really want to adopt these, but then you have the activists saying, no, you can't adopt these. We just want all your crops to die instead.
And also, if you do, then you can't export those crops to the countries that would actually pay you for them in Europe.
Yeah, well, that's true there. But, just feeding their population, it would be nice if we had a crop to feed them. And so
is that resistance also RNAi based?
Yes, it's always RNA based. And so these viruses are transmitted by either white flies or leaf hoppers. I don't know what a leaf hopper is, but white flies, you get those all the time in your greenhouse.
I think leaf hoppers are hemipterans.
Yes.
Oh, okay. And
bugs, true bugs.
Are they Hemiptera or Homoptera? I'm just looking that up.
Hemiptera, I think.
Hemiptera, yes.
There's a plane called a leaf hopper, no?
I think that's right. It's a very light plant that lands on the top of trees.
So, and do a lot of people work on Gemini viruses among plant virologists?
It's huge.
Yeah.
So it's not big in this country. There's only about 3 or 4 groups.
There's
that
a lot
work on
more than 2
Gemini
people
viruses.
working on them.
But in China, they have entire institutes devoted to Gemini viruses. And it's the major study in countries like India and Pakistan. There's major groups in Israel. Certainly Africa is really big. South Africa, they've got major Gemini virus groups there.
What about native plants that are not edible, like just, wild plants? Does that infect them too?
That I don't know. I don't know what their wild hosts are, what their reservoir, because, there's reservoirs, just like they may not cause symptoms is the thing. And so if they don't cause symptoms, you generally don't know.
Got it.
They have such lovely names like tomato golden mosaic virus and tomato yellow leaf curl virus.
They're very descriptive, very descriptive.
Yeah. That's a bad one. And so there's a lot of these viruses, and they have to. So the cool thing about these DNA viruses, so of course, as you well know, having written, the virology textbook with very little plant material.
Volume 4.
Volume 4. Yeah, Volume 4. You probably don't remember me complaining about Volume 1 because there's been Volumes 2 and 3 since then. So DNA viruses, they either replicate in the cytoplasm or they replicate in the nucleus. And the viruses that replicate in the nucleus have to, if these are not dividing cells, they have to push the cell into what's called S phase, the phase of the cell cycle where you're going to replicate your DNA. So the cool thing about plants is that very few cells are actually dividing.
Right.
So when you see a leaf, when you actually see the leaf emerging, all the cell division is over. It's just expanding at that point. So when an insect infects, a whitefly, let's say, is transmitting a geminivirus, the geminivirus now has to push the cell into S phase. So a lot of times with these viruses, you see these like cancer-like nodules on their leaves where they've literally pushing the cells to just keep dividing. So that's kind of a cancer. If there was a plant cancer, they that's what they have to do.
Well, same thing is for animal viruses because most of animal cells are not dividing, and viruses like SV40, adenovirus, they all have to push cells into S phase as well. Otherwise, they can't divide. Except the
viral oncogenes.
Except the pox viruses that, as Condit says, they avoid the nuclear bureaucracy.
Pox viruses are pretty smart.
Are they smarter than plants?
I think so, yeah.
Wait. Plants are not that bright, but are they smarter than the virus? I think so. So Gemini viruses, they make this protein called rep, standing for replication. And this protein pretty much does everything. This is this multifunctional protein. And one of the things that this protein does is it binds a protein called retinoblastoma, which is
I know that protein.
And you should, and the animal people should know that protein.
Reminded me of large T in SV40.
Yeah.
Where it's like this, it's got this one protein, but that's all it needs because that one protein does all sorts of things.
Yeah, it directs traffic for everything else, all the cellular.
Phosphofructokinase.
Well, RB, of course, keeps cells from going into S phase because it binds to a transcription factor that and it keeps it inactive. And so when rep binds to RB, it releases the transcription factor, and now the cell goes into S phase. And so the virus uses this rep protein to do that. And this paper is talking about the other things that this rep protein does, because it binds a lot of things. And one of the things that it binds is this protein called proliferating cell nuclear antigen, PCNA, which of course is in animal cells as well. It's this clamp, this sliding clamp that is involved in DNA replication. And so the cool thing about this paper is that this PCNA gets these little tiny proteins added to it called the SUMO, which is a lot like ubiquitin, which are little proteins that get added. And when they get added to other proteins, then stuff happens. The protein might now bind something else, or maybe it can't bind something else, or it gets degraded. So it's a way of kind of switching a protein from doing one thing to doing something else. And so this PCNA normally binds this little SUMO, and what rep does is it keeps PCNA from binding SUMO. And so the cool thing is that PCNA, when it binds to SUMO, that's when it does just straight DNA replication. When it doesn't bind this little protein, it does what's called homologous recombination, which is where you literally attach pieces of DNA to each other.
Right.
And this virus really loves homologous recombination. I mean, most of these 360 strains, you can trace them back to, oh, this one's got this parent, and then that one had that parent and that parent. And so the grandparents and the great-grandparents and stuff are all different viruses that have recombined. And so it's a big recombination mess in Gemini viruses. And that's because by keeping this pCNA from binding to SUMO, it makes it do recombination. And that is good for the virus. So what this virus does is it literally makes the host cell an environment that is exactly what the virus needs in order to replicate and then get transmitted from cell to cell. So this paper is the first paper that actually showed that this pCNA protein was actually sumoylated. I don't know how.
Sumoylated, I think.
Sumoylated.
So it actually gets sumos on it, and that had never been shown before in plants. And then they very, very, very clearly showed that this rep protein keeps pCNA from having these little proteins added to it. So it's just another way that these Gemini viruses can change the cellular environment to make it a happy home for it and its children.
I found it interesting that most of what they do in the paper is to produce these proteins in bacteria, right? There's one experiment at the end in plants.
Right.
Implante, I love the term.
Yes.
Implant.
Yeah, we say implant.
You guys all say implante. Is that right?
Well, yes, we actually use, so the plant viruses community and the animal virus community use different words to mean the same thing. So in our lingo, in vitro means in a test tube.
Right.
In your lingo, it's cell-free. Okay. In our lingo, in vivo means in cells. Okay. In like protoplasts, which are cells without cell walls, individual cells. So that would be like the animal cell culture system, which you say is in vitro.
Right.
Right. So your in vitro is our in vivo, and then our in the organism we say is in planta.
Right.
And you would say that was in vivo.
Yeah, we have no other word. We could say in animalia. Right?
Every grant proposal, and I tell this to the plant people as well, because your grants are going to be read by animal people if you're submitting them to NIH. Define your terms.
Yes.
I love it.
Understand what you're talking about.
So we're animal people, huh?
You are
Yes.
animal.
Well, now, I gather some of this distinction is because plants are a little fuzzy about whether they're growing in culture or growing as a whole plant, right? You can culture the plant cells and then generate a plant from them in many cases.
Yes. So you can de-differentiate plant cells. So the way what you can do is you can take, any tissue of a plant pretty much.
You mean like a stem cell?
It's almost as if every plant cell is a stem cell.
Well,
Even if
In
they're
other words,
leaf cells.
you differentiate your differentiated cells back to stem cells, and then you can make them just grow as a mass of cells. You just change the hormone conditions. So you grow a mass of cells. That's called callus, okay? And it's just this big mass. And then you can just change the hormone conditions, and all of a sudden you get leaves from your callus, or you get roots from your callus, and then you have your little plant.
It's as if you could take the L cells out of the incubator and just have a mouse pop out of them.
Exactly.
Boy, wouldn't that be great?
That would be so much easier.
Yeah.
So that's why you use these terms like in planta versus in vivo or in vitro, is because there's a different set of distinctions between these materially.
Cool.
Yeah. And since the L cell genome hardly looks like a mouse anymore,
right?
You would have quite an interesting mouse coming up from that.
Yeah. Can you imagine making humans out of HeLa cells? Whoa.
Yeah, it would be kind of like
that would be a
thing.
problem. All right. So and not only do they do most of the experiments in bacteria, but they use human SUMO and SUMO conjugating enzymes.
Right.
Yeah.
Why don't they use the plant?
Well, they did towards the end.
Well,
they used the Arabidopsis.
They use the Arabidopsis, but not tomato, which is what most of the...
Right.
Is there any reason they just don't have them cloned yet, or is this what you plant guys do, or what?
Well, what you have to understand is that there are very few of us. So much fewer. Our population is much fewer. So if you think about how many people work on the virus that I work on, turnip crinkle virus. The answer, so here's a virus, it's a major virus, major model system. And worldwide, there are exactly 2 groups that work on it, me and one other. Right.
Okay.
I mean, so that's it. So this is kind of common. So the problem is that you don't have a lot of things cloned that you do in animals. So most of what is cloned is from Arabidopsis because it's such a fast growing plant.
It's
and
an
it's
angiosperm,
such
by
a
the way.
huge
It's an-
model.
Thank you very much.
there are so many resources. It's what we call the yeast of the plant world.
Right.
Okay, let me take another
Now,
approach. You must have
in the case
genomes.
of
You must have
in the case of, I just wanted to point out, in the case of PCNA and SUMO, it really kind of doesn't matter what species you're using because these are extremely highly conserved, right?
You only need
Right.
one amino acid to make a difference.
Well, okay.
I mean, we do the same thing. I mean, we use rabbit reticulocyte lysate sometimes
Sure.
to do our, in
Right.
vitro translation work.
But Ann, all these genome sequences are known. You could easily have the cDNA synthesized, right? Or is it that you don't have enough money, you plant guys? Is that
Yeah,
part
well,
of the
yeah,
issue?
give us money. Yeah, I mean, but again, yes, you have to have it synthesized. You have to have it, working. I mean, the thing is that you can make a phone call and get the system
Mhm,
and see if it
mhm.
works. And it did work for them. And so then I'm sure the reviewers said, yes, but what about,
Right.
plants? I can't remember if the reviewers said that.
In particular,
No, actually,
because there were some differences between what they got and what some other groups got. So they really had to go try and sort that out by using the real plant genes and et cetera.
Right.
Right.
Hey, Ann, I have a technical question here. So maybe you can explain this. So they say the last experiment, we transiently expressed tomato PCNA in young, not fully expanded N. benthamaiana leaves 2 weeks. So can you explain what
Does that mean?
Yeah.
Okay, so the first thing is N. benthamiana. So this is Nicotiana benthamiana. This is the plant that is growing in virtually every plant virologist lab because this plant is just such a wonderful plant. First thing, almost every virus infects it, from saguaro cactus virus through opium poppy mosaic virus. That's another one of our viruses, by the way. We thought maybe we could get some Defense Department money if we were going to go after opium, but no such luck there. So everything infects this plant. So it's a nice plant for that, but that's not why they're using it. They're using it because you can do this technique called agroinfiltration in it very, very easily. So agroinfiltration is this wonderful bacterium called Agrobacteria tumefaciens, and it was what was used for all of the original genetically modified organisms because this bacteria naturally produces a plasmid, and on the plasmid is a little piece of DNA. And if you attach anything you want, any gene you want to that little piece of DNA, Agrobacterium will happily inject that little piece of DNA into a plant cell where it will then get into the nucleus, and for days it will express whatever you have on that piece of DNA.
Right. Is this the little plant from Australia that's related to tobacco?
It's not small. It's very big.
It's really big? Because there's another plant that I saw in Texas at a vertical farm that was using plants to produce proteins of use in medicine for people like insulin, and they were doing influenza virus capsid proteins. In this plant, it's a little
changing the nutrient value to keep it small, but this is a plant that.
What's it related to?
Okay, so just to, it's related to tobacco. It's in Solanaceae. It's in the Solanaceae family. This particular one, benthamiana, is from Australia.
Okay, so that's the plant that they're using.
And this is not the commercial tobacco that people smoke.
No, of course.
This is a different plant.
That's right.
That has become, as Anne says, the standard for plant virology.
So that was their system. The one that you're describing right now is the system they were using.
The nice thing is that it's very, very easy to do this technique because what you do is you put your agrobacterium carrying your plasmid in a syringe without a needle, and you literally push it. You go to the top of the leaf and you just literally push it in. And I guess the veins are big or something like that in this plant because it's. Yeah, or the stomates are wide open or something, but it goes right in, and it goes and it travels throughout, and every single cell gets the tDNA, and so you have a really nice synchronous system there, and not every plant can be agro-infected like this, agro-infiltrated like this.
The plants in Texas were put into a vacuum chamber, and then they made a mist of agrobacter that was genetically altered, and then
That's another way of doing it.
Turned on the vacuum and then just pushed everything right down the lid.
Pushes it right in.
It did.
Slurps it in.
So when you put a plant in the vacuum chamber, the stomata open maximally.
Yeah.
And that allows for 100% infection. Yeah.
So, Anne, you have done this to put genes into plants, right?
I do this routinely to put viruses in plants.
Viruses.
Uh-huh.
So it's just a very nice way. And also you can have, replication systems because it's very easy to express different proteins and then express your virus. And you could pretty much put 4 or 5 things, probably even more, and get them all into the same cell at the same time. And then they all get expressed at the same time, and so you have a nice little factory going on with these cells. So it's great. Wonderful plant. We love it.
So
and it has great symptoms. Boy,
they don't let people work in this vertical farm that smoke because they have tobacco mosaic.
They have tobacco mosaic.
And it would kill all the plants.
The N. benthamiana, the laboratory strain, I gather, is a particular strain that is highly susceptible to all these viruses. So people may be wondering, well, how is this thing susceptible to every plant virus and survives in the wild? And I think the answer is that the wild strains are not susceptible to every virus.
Yeah,
this particular.
They can't be. But this has a very odd RNA silencing system. So its innate immunity, the way that it protects itself against viruses, doesn't work well. Okay.
So this is skid tobacco.
Okay.
And so everything kind of will infect it. And
so in this experiment, they put tomato pCNA, all right, and they put Arabidopsis sumo into N. benthamiana, and they showed that the pCNA would get sumoylated, and when they put a rep in, it suppresses the sumoylation. So everything they found out in bacteria happens in plants as well. And in fact, it gets sumoylated on the same residues and so forth. So,
right. So that was good. I mean, and so it confirmed that the bacterial stuff that they were doing, was, made sense that they could trust it.
So I want to make sure I understand the role of the homologous recombination in this, because so the end result of this is that the virus, by altering the sumolization of pCNA, is shifting the attention of the DNA metabolism machinery toward the homologous recombination away from replication, right?
Correct.
But that's
clearly it's still replicating the virus.
Yeah, and they talk in this paper about recombination mediated or catalyzed replication, and so I get the impression that in this particular case, the recombination system is actually being used for replication. And what comes to mind is I'm thinking that in the process of doing homologous recombination, you probably generate structures that can serve as replication origins. Am I thinking about this correctly?
The replication origin on the virus, there's a hairpin that's the replication origin.
Okay.
So, it's a little bit different than your normal DNA origin of replication. Well, quite a bit different from that.
But it's not just recombination that the virus is doing with this. It's actually actual replication using recombination machinery. I guess we tend to sort of over-compartmentalize this stuff. We think of there's DNA repair over here, and there's DNA replication here, and there's DNA recombination here, but they share a lot of factors, and there's probably a lot more overlap amongst these processes than we normally think about. So it's not as if you only do recombination with recombination enzymes.
Yeah. So would I be correct in my understanding that we were switching from the recombination that goes with repair? We're turning that off in this paper and turning on the kind of recombination that goes with replication.
I mean that's the way I see it. But I'm not an expert in DNA viruses and recombination. It's just that they believe that, if you do a lot of homologous recombination, what you're doing is genetically you're producing a more varied, population of
Yes.
viruses. And apparently that's very helpful for the geminiviruses as
But
well.
they shouldn't interfere with
Because the DNA polymerases don't make, very many mistakes. But this way, with homologous recombination going on, you generate more diversity.
So, I mean, my interpretation is, as Rich says, that this is important. This recombination system is important for replication because the long-term survival would depend on recombination. But if you're just doing an assay for DNA replication,
Right, it has to replicate.
it has to replicate. And so the recombination system must be essential for that, right? Because allowing pCNA to be sumoylated, if I understand, inhibits viral DNA replication.
Correct.
So that's
Strongly.
it's an immediate
Yeah.
effect, right?
Well, and the thing that I noticed, and obviously this is coming from me as an animal person who works on DNA sensing. Yeah. Is that we have a lot of ideas about how innate immunity to DNA is related to repair. And so there have been quite a few papers recently about this. And so I was thinking, so maybe we're avoiding repair so we avoid innate immunity. And then I fell down the Google hole of DNA sensing in plants and trying to understand the plant innate immune system. Yeah.
Yeah, very different. So I'm sure that this is what the group is going for, is, exactly why, how is this desumulated, pCNA able to preferentially replicate the virus, because it really doesn't want it replicating the genome. I mean, it wants to use all of the components to replicate itself.
So actually, I see there's a last sentence in the papers.
Exactly.
Besides its effect on increasing Gemini viral recombination, the higher homologous recombination activity could also have an effect on replication efficiency, since homologous recombination provides a mechanism for tolerating lesions that could block progression of replication.
Yeah, so that's a theory. That's good. Yep.
That's a theory.
Yep.
Yeah.
That makes sense. Okay. Yeah, it has to be an immediate effect, because recombination clearly in long term is
long term.
That's what I was thinking.
Yeah. Good. All right. So is this a typical plant virus paper, Anne?
Yeah. I mean, it's a lot of times we're trying to do in plants what has been done in animals, or we're checking to see if there's similar things going on, but certainly in the fields of replication, translation, things like that, I mean, the papers are I think we're really leading the virology field. And so I don't want to blow my own horn here.
One of my take-home messages from the discussion today and the discussion we had at the ASV meeting is how important these viruses are for us as humans, for the planet, because they really have a profound effect on our food supply.
But remember, these are all vector transmitted viruses. Growing them indoors will solve that problem.
Yeah,
That's correct.
But as we said before, you can't make trees indoors.
No, but you can grow cotton indoors. You can grow soybeans indoors. You can grow all these other plants indoors.
Yes,
Until a few bugs get into your vertical farm.
And by the way, there is a crossover plant that probably will confuse all of the listeners right now, and that's the tiger lily. Animal plant.
Yes,
Yeah.
Sorry.
Is that, Alan, is that like bugs getting into your software?
Into your software, I assume so.
Ooh.
All right.
Yeah, I mean, viruses cause
huge economic loss.
losses worldwide.
Any estimates?
And I mean,
billions and billions.
Yeah, because
billions. It's probably in the hundreds of billions.
The plants that are left standing are probably eaten by insects. So
right.
We don't get anything.
The worst are the trees. I mean, that's just so sad, because then they're just lost, and 100-year-old trees are lost. So viruses are pretty devastating pathogens. So trying to understand them is important. And of course, we need more groups working on plant viruses, and we need reviewer 3 on your NIH grants to stop giving you negative comments because it's not animal virus pathogenesis. Are you listening, reviewer 3 out there?
So how supportive is the USDA for plant virus research? They have a research group.
Yeah, are they
Crickets.
listening? The problem with USDA is that it's a problem for people like me, is that they concentrate on pathogens of important crops.
Right.
And of course they should be concentrating. Most people have
that's right.
But of course they should be concentrating on important pathogens of crops. Just like, of course, NIH should be concentrating on important pathogens of animals and people, and not like, coronaviruses that are pretty much, eradicated.
It's just one of them, not all. There are a lot of others.
That's true. So,
but there are other viruses out there too.
There's model systems,
Yeah,
and so
of course.
USDA does not support model systems anymore. So they don't support
That's too
model
bad.
viruses, and they don't support model crops.
In their defense, they don't have huge quantities of money
And they
to
have
throw
very
around.
little money, and they have devastating problems. As I say,
I mean, I should say they have a lot of money that goes to other aspects of their many branches of their mission.
Have you ever seen the
they're
Farm Bill?
they're making sure that beef that says grade A is grade A, and they're making sure that the stuff in the supermarket has ingredients lists on it. They're making dietary recommendations and yada yada yada, and then we do a little research too.
Yeah, but the Farm Bill that's passed every 5 years is over $300 billion.
Most of which is a welfare program. Yes,
half. I would say
Yeah.
half.
So
I want to.
they're not the ones supporting the plant viruses. So
What
who
a
is
shame.
supporting the plant
Yeah,
viruses
right.
is NSF.
right.
And the National Science Foundation has been phenomenal. They're the ones that supported people who first started working on RNA silencing. They're the ones they've supported me from the very beginning of my career. And they support model systems. They don't have panels for virology. I mean, you're in a regular panel, molecular cellular biology panel, but they have plant virologists on the panel, and they're very supportive because they know that by studying these viruses and by studying the RNA and RNA structures and RNA structure function, things like that, that we're making huge strides in understanding, cellular RNAs. And as we all know, a lot of things were first discovered in viruses and then later discovered in cells. And so they've really put money into plant viruses, not as much animal viruses, because they know that NIH is supporting animal virology.
Right.
So kudos to NSF for doing this.
By the way, the third reviewer, it's a problem for us animal people too.
Yeah.
It's a problem for all of us.
I want to fill out the acknowledgments for this paper because I think we acknowledged Eduardo Bejarano.
Yeah, go ahead.
The first author was
Manuel
Arroyo Mateos, and then
there were a lot of. And can I also say something about plant virus research in Spain and in the Netherlands, where the groups were that did this work?
Oh, the Netherlands
they have
is good.
Wonderful plant virology.
Yeah, they're great.
And I mean, Spain is just one of the top places in the world for plant viruses. Superb research going on there, well supported. Of course, I'm sure they would like more money too, but and the Netherlands as well. The Netherlands was a real powerhouse for plant viruses. Not quite as much right now, but certainly they have been over the years.
All right. Anne, do you want to...
Yes, I do have to go, but I've really enjoyed being here for TWiV, and I'm looking forward to a monthly TWiV with all of you guys.
Yeah.
Great. Look forward to it.
All right, Anne
I'm looking forward to learning.
Anne,
Well, thank you for having me on.
my pleasure. Bye.
Bye.
By the way, Anne is from the University of Maryland.
Yeah.
And on Twitter, she's Anne, ANNEALIZ1. You can go check her out. Let me finish the emails from before. Let's see. There was one prion bit from Nicholas. Chronic wasting disease sometimes is covered on this show. I am aware that there are no known human cases associated with chronic wasting. Has anyone investigated if coyotes or wolves might have caught the disease? My reasoning is that these animals are more likely to eat the entire deer. This distinction is important because if I remember correctly, Kuru, a human prion disease for the uninitiated, was only spread by person to person because individuals eat the brains of the infected deceased. Surprising to me that only deer have been documented to suffer from this prion disease, considering how so many different animals like to eat them. However, a possible answer is simply that a prion disease takes a long time to manifest, and the predators that eat deer generally do not live long enough for CWD to develop in these animals. So, if I understand, it's cervids that develop it,
Right.
deer, moose, elk.
Yep.
I don't know about their predators, though. That's a good question. You know,
The places that CWD has been observed are generally places that have very few of these predators, because it's not true that a lot of different animals like to eat deer. They actually have only a few natural predators, which have mostly been extirpated from the eastern half of the United States.
Like cougars.
Like cougars and wolves, and that's pretty much it.
Right.
Every now and then a coyote will get a small fawn.
Yeah.
Or maybe a very, very sick older deer.
Don't forget roadkill, though.
There's certainly roadkill.
There's tons of it, in fact.
But that's that's going to be eaten by a variety of scavengers over a period of time until it gets scraped up. And I'm not aware of any data on wolves that have eaten deer getting CWD, but it may just be a case that there hasn't been an overlap that resulted in a visible case, because we have so few wolves and so few cougars, for example.
Mm-hmm.
It's interesting. I have an article here from the Timber Wolf Information Network saying that researchers say wolves could help curb wasting disease in deer.
By eating the sick ones. That's
yes,
right.
exactly. So they don't mention that they might get it.
Right.
They say they might get rid of them because, that's what wolves do. They go after sick animals.
Yes.
Okay.
And that's why we have a deer overpopulation problem in the built-up areas in the eastern U.S., because we got rid of the natural predators and we didn't replace them.
Now, the Toyota is the natural predator.
Well,
Yes, but it's not a very effective one.
Not really.
No.
All right, let's do one round of email.
Okay.
All right. Are you queued up there, Dickson?
I'm queued.
Can you get that first one from Bob?
I can. Bob writes, still on TWiV 508.
Wait a minute. What are you reading? What do you
Bob writes. You asked me to read Bob?
Go down to the email section that says finish email
Bob
above first.
Writes.
Well,
and then Theodore writes.
You want to you want
which one do you want?
Bob.
Got it. Okay. Although this article, and then he gives the article from medicalexpress.com, is hugely interesting in its own right, I thought for dedicated anti-vaxxers, this statement in it was especially noteworthy. Quotes, when you're fighting bacteria that can double in number every 20 to 30 minutes, every moment matters. To put it bluntly, if your immune system takes too long to assemble the tools to fight the infection, you die, he says. Regards, Bob. And have a nice day.
Well put.
It's a very interesting article. I talked about it a little bit with one of my classes maybe 2 weeks ago, and it is really a cool article.
Yeah, the problem with anti-vaxxer literature is that the anti-vaxxers don't read it.
Yeah.
Well, this is not directed at them, but he happens to. So tell us about this briefly, Brianne. What is it?
So basically, the idea is that some new structures in the lymph node were described. They're called the subcapsular proliferative foci, SPF. And if you read some of the press about them, it will say things like, immunologists discovered place where all of immune memory comes from, or, really, really, underselling it there.
Yeah.
But it basically looks like there are structures that go across entire lymph nodes that allow for proliferation and location of memory cells.
Yeah, cool. Yeah, I think we have to do that on immune sometime. All right, Alan, could you take the next one, please?
Sure. Theodore writes, today's Sun Sentinel newspaper had this picture of a VW Beetle decorated with various cell paintings and this mega virus on the roof. Science writ large. I was unable to Google any information about the artist or owner, who undoubtedly is a scientist. And that's from Ted, our friend at Nova Southeastern University in Fort Lauderdale. And this is he's got a shot of a newspaper. It's a VW Beetle, new Beetle, that's gotten a graphic wrap with, I guess that looks like some kind of cells floating in solution or stylized ones. And then on the top, there's this, I'm going to guess that's an enveloped virus, and those are glycoproteins sticking out from it. This huge thing taking up the whole roof of the bug.
I want to put virus stickers on my car. I'm going to look and try and get them. I think that would be so cool. I'd love to cover my car in virus stickers. No, you don't like that, Dickson?
Well, there are shops that will do an entire wrap like this. I don't know what that costs. I think it's usually done for promotional vehicles.
Oh yeah, they'll wrap it.
Yeah, I think this is usually done with a sheet of vinyl that's stuck to the outside. And the nice thing about it is that you can then peel it off when you go to sell the car or trade it in.
I'm going to look into it. I want a wrap covered with viruses.
There you go.
That would be so cool. I love it. I love it. Rich, can you take the next one?
Sure. Stephen writes, I just listened to TWiV 508. Very interesting episode. Here's an Amazon link to a book written by one of the defectors mentioned on the podcast. So 508 was when we talked to Jens about biodefense and bioweapons. And so, the link is to Biohazard, the book Biohazard by Ken Allebeck.
Right. Yep.
And let me see. Does he have another one here? Oh, yeah. There's a wiki link to Ken Allebeck. There you go.
Why don't you take the next one, too?
Bob writes, still on TWiV 508, during Jens Kuhn's exposition, the question came up of why Hitler did not have Germany embark on biological weapons work. I wondered whether that was because he was gassed in his World War I service. For example, see this Wikipedia article, and he gives the Wikipedia article. He gives the link and then quotes from that article. On 15th of October 1918, he and several comrades were temporarily blinded, and according to Friedland Wagner, Hitler also lost his voice due to a British mustard gas attack. After initial treatment, Hitler was hospitalized in Pasewalk in Pomerania. While there on 10th November, Hitler learned of Germany's defeat from a pastor, and by his own account, on receiving news, he suffered a second bout of blindness. Regards, Bob. I seem to recall in reading the material that Jens sent us that there was something about this, that as a result of his experiences during World War I, that it was those experiences that turned him against chemical weapons.
Chemical and biological weapons.
Yeah, right.
He also
he was a vegetarian. He was a weird dude.
Yeah. Also, the Third Reich spent a lot more attention to technology development than they did to science, which is why they had tremendously effective stuff at the beginning of the war. And then when American science got mobilized and we started outpacing them with things like radar and the proximity fuse and all this, they weren't able to keep up.
Right.
All right, Brianne, can you take the last two?
Sure. Ken writes, greetings, TWiV team. And then he gives a link that describes a study that reveals that many Twitter posts about vaccines were generated by bots and Russian trolls. So it's a pretty interesting link. Maybe, all of our anti-vaxxers aren't coming from where we originally thought.
Right.
Yeah, we talked about this, I think, a couple of TWiVs ago.
Mm-hmm. I think so, too. And then Anthony writes, I've not thought much about planaria since high school. There appears to be one species prominent as a model organism. I remember we talked about this with a paper recently. Schmidtea mediterranea. And he gives a link and also to work that has been done on that species. There is a species that is a pest in saltwater aquaria. There's another link. I don't see that any sequencing has been done on them. Perhaps you can get those from the Coney Island Aquarium or posting a request on a Reef Facebook group, Brooklyn Aquarium Society Facebook group. That planaria learned through cannibalism is questionable. Something more solid is that if a trained planaria is cut into horizontally and then allowed to regenerate, the former tail end retains memory. For what it's worth, Anthony.
I wonder if it's infected with viruses and you cut it in half, which half gets the viruses? Or do both halves get it? I'd like to get some planaria and play with them for sure. That's on. All right. Let's do some picks. Brianne, what do you have this week?
So I originally found out about this from an article from Carl Zimmer in the New York Times and from Ed Yong in The Atlantic. They were both covering a PLOS biology paper that came out this week. And this paper takes a look at the 20,000 genes in the human genome and what percent of them have been studied, what percent have papers written about them, and things like that. And in fact, they say that a relatively small percent of the genes that are out there have actually been investigated. So they see that, for example, 5% of all genes account for half of the publications. And I think it was something like, 2,000 of the 19,000 genes have actually really been studied well. They come up with some reasons for that. They have to do with whether the gene is in model organisms, whether there are reagents available, sort of like you were talking about earlier with Anne, whether there's a fair amount known about them. And they say that, originally people thought with the human genome sequencing, we would learn so much, but people seem to stick with the same genes. At this rate, it would take 50 years to know sort of a amount of information, a decent amount of information about all of the genes in the genome.
And money.
And money.
A lot of those genes are similar to other genes that have already been studied, though. Did they include those into that?
They looked at
like housekeeping
genes individually. Genes. They did not include that. One thing that I thought was sort of interesting in the Yang article is that he notes at the end that they highlight some genes that had, as he said, escaped the doldrums of obscurity and become newly popular. And one of them is SAMHD1, which we talked about last week.
Right. And for me, the interesting, what I take away from this is that there is a tendency for a lot of genes to be sort of famous for being famous.
Exactly. Yes.
That people work on them
genes.
because people are working on them, and a lot of that the authors of this article trace back to they were easy to work on in the 80s and 90s.
80s and 90s. Yep.
When all this was kind of being developed, and so people continued working on them, and now there's a vast literature, and the problems are centered around those particular genes, and that model has persisted when in fact we probably have a lot of interesting stuff yet to discover on genes that people just haven't been paying attention to.
Right. And they sort of say that might be a reason why, the genetics of certain human diseases are still unclear.
Right.
Well, the way I look at it is there's a limited amount of money. You just can't pick a gene that seems interesting and work on it because you won't get funded to do it. I mean, there's tons of genes.
Famous for being famous.
Exactly.
Right.
And so that's the way science is being driven, right? By the funding. You would like it to be driven purely by curiosity, but it's not. And it's too bad.
Yeah, it's too bad there's no institute for just studying the human genome, regardless of whether you think it's interesting or not. Yeah. And let them just work their way through
Institute?
it. Something like that. And I do believe we have one already.
Yes, I think we do.
And why don't they do that then?
They don't have a lot of money.
They don't have a lot of money. That's right.
Cool. That's really neat. It's interesting that it got press,
Yes, exactly. Yeah, I saw both the Zimmer article and the Yang article right away on the same day that the paper was published.
Yeah, it's not surprising at all.
Yeah, that's the magic of embargoed press releases.
Yeah. Alan, what do you have?
I have an essay in the New England Journal of Medicine that is open access. Most of the NEJM is not, but you can go and read this essay, and it is by a former TWiV guest about another former TWiV guest.
It's right.
So this is Judy Lieberman writing about Joan Stites, and Joan Stites was in the news because she just received a very prestigious award, the Lasker Koshland Special Achievement Award in Medical Science. And that award was for, of course, Joan's pioneering work on RNA and also, incidentally, for her work as a mentor.
Cool. That's
and
great.
Very cool.
Judy, of
Very
course,
good.
has had very similar, path and of the same generation, and is an excellent person to write this article.
Yes, and in fact was the mentor for my first rotation.
Oh,
Nice.
Rich, weren't you involved in that also?
Yeah.
Joan was my PhD mentor.
That's exactly what I thought.
So this is so good. If we do TWiV long enough, eventually we'll
They'll all
cover
be on.
everybody.
yes.
So Kathy earlier I guess shortly after this award was done, sent me a tweet from Akiko Iwasaki, who's an immunologist at Yale, who tweeted, I guess, as she heard or right after she heard a talk that Joan gave at Yale, accepting her on the occasion of this Lasker Award. And the tweet was basically that all Joan talked about was she never said anything about herself, about all the help she got and all the support she got, and how grateful she was for all the people who helped her do her work. It was just incredibly generous. So that's cool.
And I had her as my keynote in 2015, ASV.
Yep. Nice.
And we had her on TWiV. It was great. Rich, what do you have for us?
Okay, I have a CNN article that I found that, I think everybody's probably aware of this mystery, but it needs drawing attention to. The CNN article starts off talking about Richard Feynman and how the one problem that Feynman couldn't solve was how come if you bend a piece of spaghetti until it breaks into more than two pieces.
That's an important question, kids.
It turns out to be an important question. And the CNN thing goes on to describe how this has now been solved. And it references a PNAS article that was published that basically says that if you twist the spaghetti as you bend it, you can make it break in two pieces. And the deal is that when you bend it and break it, the breakage transmits a bunch of energy to other parts of the strand, and that's what causes it to break in multiple pieces. If you twist it first and then bend it and break it, that energy is released through untwisting it rather than breaking it again. And the paper is terrific. They have movies that the paper's behind a paywall, I believe, in PNAS. The supplementary material, I don't know, but they have movies that shows their experiments, and they actually made a spaghetti breaking machine to do this. But it sounds a little silly, but it has all sorts of applications. You wouldn't believe the math in the paper. It's crazy. Right.
So I think we should note here, though, that PNAS should stand for pasta and anchovy sauce.
And just the last bit of this article, the last sentence, as he said in his best-selling book, Surely You're Joking, Mr. Feynman, Feynman's reasoning behind picking the problems he sought to solve, quote, didn't have to do with whether it was important for the development of nuclear physics, but whether it was interesting and amusing for me to play with.
Right.
Cool.
Right, which also reminds me of the fact that Einstein received his Nobel Prize for studying Brownian motion, which he thought was interesting. Why? We don't know. But it was interesting. Still is interesting.
So what do you have?
Well, I haven't seen this movie yet, but I picked it because I saw the coming attractions.
Did you read the paper?
Stop it. Have I ever participated in a science fair? Was I ever a card-carrying member of a... No. The fact is that this celebrates science, and it celebrates the enthusiasm of young people who glom onto science for the first time in their lives and the encouragements that they get. It's a documentary film. It's a full-length feature film, and I'm going to go see it. I know I will. But the coming attractions were just good. Right. You see the beaming faces of these kids after they win something. Even the losers are joyful because of the participation. I think it's it will be a great movie. When I see it, it will be terrific. I can come back and tell you all about it.
Dickson, if it's not, will you unpick it?
Sure. I will unpick this, but it got a good review, and I think that we can count on the fact that this is a good movie.
Sure. Cool. All right, my pick. I'm sorry I have picked this on TWiV the other day, but I want everyone to hear about it. This week I went to Mount Sinai where they had their first PhD lab coat ceremony. As everyone knows, medical students, when they come, they get a white coat ceremony, get a coat, you get the Hippocratic Oath. PhD students are ignored. Mount Sinai wanted to change it, so they had a ceremony, which was really nice, and they asked me to do the keynote, so this is a link to my keynote. But more, I want people to think about doing this at your institution, because those students, they got up on stage, they got coated or encapsulated, and they loved it. Parents were there, great talks, and inspiring. Wow. So I think it's really cool.
Cool.
Wow.
Good job, Mount Sinai. A couple of listener picks. Stephen, it's getting to be that time of year. He sends this wonderful rant by Mark Crislip, Flu Vaccine Myths. It is, it was published some time ago, but it is just an epic rant about dumbasses from Mark Crislip. It is just great. For example, the vaccine gives me the flu. Dumbass. It's a kill vaccine, etc. Just read it. It's really, really, really good. And then we had some picks up above that I saved.
Yeah.
Let's see. Who gave a pick? Eric didn't have a pick. Jolene. Yeah, my listener pick is the article series called Research Matters, published in both PLOS Pathogens and PLOS Biology, in which active scientists speak directly about why basic research in their field matters. I like them because they're short and tell the people's stories behind why basic research is so important. And then we had a listener pick from Nicholas. For exercise enthusiasts on TWiV, there's an app called Charity Miles. When you walk or bike a certain amount of miles, the app will donate money, about $1 every 4 miles, to a cause of your choice. One of the charities is Shot at Life, which provides measles vaccines to children who cannot otherwise access them. $1 every 4 miles, some of you might scoff. Well, as many are aware, vaccines are an excellent investment, so even if you are only putting in $1 towards this cause, the economic benefits are around $44 in return. This is not to mention the importance of protecting the most vulnerable from preventable diseases. I hope that this app gets the TWiV bump. And there you go. Bump away. All right, that's TWiV. That's good. TWiV 512, you can find it at microbe.tv and wherever fine podcasts are distributed, like on your podcast player on your phone or your tablet. Please subscribe so you can get every episode, and the numbers will help us. Send your questions and comments, TWiV at microbe.tv, and consider supporting us, microbe.tv/contribute. Our guest today, no, she's not a guest, our monthly, participant.
Yes.
Was Ann Simon from the University of Maryland, who is left to go riding on her horse.
Indeed.
She does
plant
every
co-host.
Day. Our plant co-host. In plante vive. Thank you, Ann. Dickson Despommier is at Racaniello.org.
Yep.
Thank you, Dickson.
Up and running.
The newly revised Racaniello.org.
Yeah, thanks to you.
Brianne Barker is at Drew University over in Madison. She's also on Twitter as BioProf Barker. Thank you, Brianne.
Thank you. It was a lot of fun to be here today.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at turbidplaque.com, and you can find him on Twitter as Alan Dove. Thanks, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support and Ronald Jenkins for his music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.